The hypoglossal nerve then travels deep to the hyoglossus muscle, which it supplies. It then continues and supplies the genioglossus muscle.
Neurons of the hypoglossal nucleus are derived from the basal plate of the embryonic medulla oblongata. The musculature they supply develops as the hypoglossal cord from the myotomes. The hypoglossal nerve provides motor control of the extrinsic muscles of the tongue, genioglossus, hyoglossus, styloglossus, and the intrinsic muscles of the tongue. The hypoglossal nucleus interacts directly with the reticular formation, demonstrating the automatic and reflexive control of tongue movements.
Cranial nerves are numbered based on their position from front to back of their position on the brain.
Cranial nerves have paths within and outside the skull. The paths within the skull are called intracranial, and the paths outside the skull are called extracranial. Grossly, all cranial nerves have a nucleus. With the exception of the olfactory nerve and optic nerve, all the nuclei are present in the brainstem. With the exception of the olfactory nerve and optic nerve, the cranial nerves emerge from the brainstem. The oculomotor nerve and trochlear nerve emerge from the midbrain. The trigeminal ganglia of the trigeminal nerve, which occupies a space in the dura mater called Meckel's cave. Additional ganglia for nerves with parasympathetic function exist, and include the ciliary ganglion of the oculomotor nerve 2, the pterygopalatine ganglion of the maxillary nerve.
Location nerve. Cribriform plate. Terminal nerve 0.
The glossopharyngeal, vagus, and accessory nerves exit the cranium via the jugular foramen. The terminal nerve, or 0, is a thin network of fibers associated with the dura and lamina terminalis, running rostral to the olfactory nerve. The olfactory nerve passes through perforations in the cribriform plate. The optic nerve passes through the optic foramen in the sphenoid bone. The maxillary division of the trigeminal nerve passes through foramen rotundum. The mandibular division passes through foramen ovale. Emerging from the jugular foramen, the glossopharyngeal, vagus, and accessory nerves control critical functions in the throat and larynx, including swallowing and speech.
This area here is what we call the cerebellum. As you can see, there are no signs of acute issues.
And the darker areas.
I've noticed my hands shaking a little, especially when I try to write or type. It's worrying me.
I understand your concerns. We'll schedule a few tests to see what might be causing this.
As you can see on the MRI here, the disc at L4, L5 is significantly compressed, which is causing the pressure on the nerve.
A tumor is an abnormal growth of body tissue. In the beginning, tumors can be non-cancerous, but if they become malignant, they are cancerous.
Nervous tissue is made up of different types of neurons, all of which have an axon. An axon is the long stem-like part of the cell.
Neurons are cells with specialized features that allow them to receive and facilitate nerve impulses, or action potentials, across their membrane. Sensory neurons relay sensory information in the form of an action potential from the PNS to the CNS. Motor neurons, or efferent neurons, relay an action potential out of the CNS to the proper effector. Multipolar neurons have 3 or more processes coming off the soma tour. They are the major neuron type in the CNS. Pseudo-unipolar neurons? Sensory neurons that have one process that splits into 2 branches, forming the axon and dendrite. Bipolar neurons? Sensory neurons that have 2 processes coming off the soma, 1 dendrite and 1 axon. Neuroglial cells are classified as follows. Microglial cells. Microglia are macrophage cells that make up the primary immune system for the CNS. They are the smallest oligodendrocytes, CNS cells with very few processes. They form myelin sheaths on the axons of a neuron, which are lipid-based insulation that increases the speed. Astrocytes, star-shaped macroglial cells with many processes found in the CNS. They are the most abundant cell type in the brain. Enteric glia, found in the enteric nervous system, within the gastrointestinal tract. The 3 layers of connective tissue surrounding each nerve are endoneurium. Each nerve axon or fiber is surrounded by the endoneurium. The 3 layers of connective tissue surrounding each nerve are perineurium. Each nerve fascicle containing one. The 3 layers of connective tissue surrounding each nerve are epineurium. The epineurium is the outermost layer of dense connective tissue enclosing the peripheral nerve.
Gray matter is composed of cell bodies, dendrites, unmyelinated axons, protoplasmic astrocytes, satellite oligodendrocytes.
Classification of tissue. In the central nervous system, white matter is composed of myelinated axons, fibrous astrocytes, myelinating oligodendrocytes, and microglia.
The function of nervous tissue is to form the communication network of the nervous system by conducting electric signals across tissue.
Clinical significance. Tumors. Neoplasms in nervous tissue include gliomas, oligoastrocytoma, choroid plexus papilloma, ependymoma.
Axons may bundle into nerve fascicles that make up the nerves in the peripheral nervous system. In the central nervous system, bundles of axons are called nerve tracts. The soma is the body of the neuron. As it contains the nucleus, most protein synthesis occurs here. The nucleus can range from 3 to 18 micrometers in diameter. The accepted view of the neuron attributes dedicated functions to its various anatomical components. However, dendrites and axons. The giraffe's neck contains a single axon that stretches several meters, illustrating the remarkable length of this neuron. Fully differentiated neurons are permanently postmitotic. However, stem cells present in the adult brain may regenerate functional neurons throughout the life of an organism. Afferent neurons convey information from tissues and organs into the central nervous system and are also called sensory neurons. A neuron affects other neurons by releasing a neurotransmitter that binds to chemical receptors. The 2 most common neurotransmitters in the brain, glutamate and GABA, have largely consistent actions. The distinction between excitatory and inhibitory neurotransmitters is not absolute. Rather, it depends on the class of chemical receptors present. However, neighboring target neurons called ON bipolar cells are instead inhibited by glutamate because they lack typical ionotropic glutamate receptors. Parvalbumin-expressing neurons typically dampen the output signal of the postsynaptic neuron in the visual cortex. Neurons can be classified according to their electrophysiological characteristics, such as tonic or regular spiking, phasic or bursting, and fast spiking. Acetylcholine is released from presynaptic neurons into the synaptic cleft. It acts as a ligand for both ligand-gated ion channels.
Glutamate is one of two primary excitatory amino acid neurotransmitters, along with aspartate.
NMDA receptors are another cation channel that is more permeable to K2+. The function of NMDA receptors depends on glycine receptor binding.
Metabotropic receptors, GPCRs, modulate synaptic transmission and postsynaptic excitability.
Dopamine is synthesized from the amino acid tyrosine. Tyrosine is catalyzed into levodopa by tyrosine hydroxylase, and levodopa is then converted into dopamine. Serotonin can act as excitatory or inhibitory. Of its 4 5-HT receptor classes, 3 are GPCR and 1 is a ligand-gated cation channel.
Purinergic neurons, ATP. ATP is a neurotransmitter acting at both ligand-gated ion channels and GPCRs.
Histaminergic neurons, histamine. Histamine is a monoamine neurotransmitter and neuromodulator.
Synapses can be excitatory or inhibitory, either increasing or decreasing activity in the target neuron, respectively. When an action potential reaches the axon terminal, it opens voltage-gated calcium channels, allowing calcium ions to enter the terminal.
They can also get modulated by input from the environment and hormones released from other parts of the organism.
By inserting electrodes into the squid giant axons, accurate measurements were made of the membrane potential. Thin neurons and axons require less metabolic expense, but thicker axons convey impulses more rapidly. The myelin sheath in peripheral nerves normally runs along the axon in sections about 1 millimeter long, punctuated by unsheathed nodes of Ranvier. Receptors respond in different ways to stimuli. Slowly adapting or tonic receptors respond to a steady stimulus and produce a steady rate of firing.
Other receptor types include quickly adapting or phasic receptors, where firing decreases or stops with a steady stimulus. Examples include skin.
Like what you share.
Noir plasma chrome bleed. Unlocking knowledge, inspiring innovation, empowering curious minds.